Hey, AP Bio. So we just want to go over a couple of the um, aspects of the review sheet that we put on Schoology that covers biochem, and also just put you guys at ease with some of the things you need to know. This first part here is just pretty much standard wording for AP Bio. Okay, we already know carbon moves through the environment. We know that it gets cycled. We know that it is used to build carbohydrates, proteins, lipids, nucleic acids. Okay. And we know that organic compounds consist of carbon and hydrogen. In addition, they also have nitrogen and phosphorus and a little bit of sulfur, especially when we are talking about proteins. Okay. We also know that living systems need water to live, and we know that the properties of water result from it being polar. Right. The oxygen has a high electronegativity, and that contributes to the polarity of water. And water being polar allows it to hydrogen bond. And that hydrogen bonding contributes to these properties that water has. Two of the big ones are cohesion and adhesion, right? Cohesion is when the same type of molecules stick to each other. So this is what causes things like surface tension to happen. This also allows water molecules to move in a column, such as they do during transpiration. Adhesion is where two different substances adhere to each other or bind to each other through hydrogen bonding. Water can hydrogen bond with other surfaces, things like glass, soil, cotton, tissues of plants, and that's because those materials have a little bit of polarity to them. So that enables hydrogen bonding to occur. This is going to result in capillary action, where water molecules kind of tow each other upwards against the force of gravity, such as we see in soil and in thin glass tubes. But this is also what causes transpiration, right? Transpiration, the movement of water upwards from the soil towards the leaves and then eventually evaporating from the leaves, is the result of cohesion and adhesion, okay? Where the water molecules are adhering to the tubes of xylem, and they're also cohering to each other, and they move upwards in a column, okay? Now, one of the things that contributes to transpiration is the process of evaporation. Water has a very high specific heat, which means you need a tremendous amount of heat added to it in order to raise the temperature by 1 degree Celsius. So 1 gram of that substance, 1 degree Celsius. So water can absorb or release large amount of heat energy, and its actual temperature doesn't change. So in order to get water to evaporate, you have to put in a tremendous amount of heat energy to get it to change phase and go into a gaseous form. And when you do that, you disrupt the hydrogen bonds, and that disruption of the hydrogen bonds leads to a change in phase. Water is also a really good universal solvent. It forms hydrogen bonds, and so it's going to be very good at dissolving substances that are charged or polar, but it's very poor at dissolving nonpolar substances. So things like lipids do not react with water. And that's part of the reason why we have phospholipid bilayers forming membranes, because those membranes have a nonpolar region where the tails are, and they repel water, and they point inwards away from the water. And this is what creates barriers. Okay. We also see that water can have a high heat of vaporization. Okay, and again, this creates water vapor, and we know that water vapor, when there's a lot of water vapor in the atmosphere, that creates kind of a global blanket, and that helps to kind of keep heat in. And that's the reason why an increase in moisture in the atmosphere, on hot, humid days, it feels even more warm, and on humid nights during the summer, it stays warm because that moisture in the atmosphere traps the heat and keeps it in the lower levels of the atmosphere. Then we have the heat of fusion, which is again for water is very high. In order to convert it from a solid into a liquid, a tremendous amount of energy has to go into it. Water also has a really high thermal conductivity. And what this means that water heats up very slowly, and it also heats up very evenly. And this is the reason why it takes a long time for the ocean temperatures to warm up and cool down, because they conduct heat very well, and it also holds heat very well. That's the part of the reason why we use water as a coolant, because it has the ability to hold a tremendous amount of heat, and then when it finally evaporates, it releases that heat and it cools us off. Okay. Um, some of the more common molecules. So here we go. Okay, macromolecules. The term macromolecule means that you're talking about a substance that's made up of monomers. So always equate macromolecule with polymer. Okay. So nucleic acids are polymers, micromolecules that are made up of nucleotides. And we did this not too long ago. So you guys remember, the structure of DNA. Okay, it's made up of nucleotides. You have your phosphate, your sugar, and your base. Okay, and in order to put a DNA molecule together, you have to have each side being anti-parallel, right? So one side runs 3' to 5', and the other one goes 3 to 5 in the opposite direction. You can see how these sugar molecules are upside down, and that's to ensure that the bases pair up with each other. Okay, the bases have hydrogen bonds between them, so individually weak, collectively strong. Okay, and the backbone, the phosphate sugar backbone, that contains the phosphodiester bond, very strong, stable, covalent bond that provides the lateral stability for the DNA molecule. Okay, and we'll get more into 3', 5' when we talk about DNA replication. Proteins, very large group of compounds. The order of amino acids in a polypeptide, that's the primary structure. What determines how the protein folds are the R groups. Okay, remember we did those activities with the tubers and we put the little magnetic side chains on and then we folded the tubers into all these different structures. Okay, it's those R groups that determine the structure and the function of the protein. Okay, some of those R groups are hydrophobic, so they're going to tuck themselves on the inside of the protein to stay away from the water. Some of them are hydrophilic, and they like to fold themselves outward to be exposed to the aqueous environment that we find in most cells. Okay, but with protein folding, the shape of the protein is going to determine its function. And that shape is dictated by the sequence of amino acids. So if you change the sequence of amino acids, we change the shape of the protein. And if we change the shape of the protein, we're altering its folding pattern, and that alters its function. Okay? And you guys may remember proteins can be things like carriers in the cell membrane, antibodies, hemoglobin, enzymes, a good chunk of hormones. We just talked about that, right? They're very large, they're globular. Okay, and when we look at the structures, right, this is how proteins are organized. Primary structures is simply this long chain of amino acids. Okay, it's just a single polypeptide chain. Secondary structure, that polypeptide chain begins to coil. Okay, and that's the result of hydrogen bonds that are occurring between the amino acids. Okay, and these little dotted lines represent those hydrogen bonds. Same thing happens with a beta sheet. Okay, except the difference is instead of the hydrogen bonding occurring in the plane of the protein, now it's occurring laterally. Okay, and so we get this side-to-side interaction here, and that results in this folding of the protein. It looks like an accordion. It's called a pleated sheet. Tertiary structure contains both of these. Okay, it contains both alpha helices and beta sheets. Okay, and these are globular three-dimensional proteins. Okay, that have all kinds of little pockets in them. They have catalytic sites. They have regulatory sites. And then if you take one of these tertiary proteins and you pair it up with others, in this case, this is forming a very large structure. This is a quaternary structure. It's made up of 2 or more polypeptide chains. So in this case, we have hemoglobin, right? And hemoglobin is made up of 2 beta chains and 2 alpha chains. And in the middle here, we have the heme group. The heme group contains iron, and this heme group with this iron in it is known as a prosthetic group. That's a group of molecules made up of something other than protein. And in this case, it's got iron in it, and that's what's carrying the oxygen. Okay? If we alter the structure of this, we alter its function. And that's what happens with sickle cell anemia. Right? With sickle cell, we change one of the amino acid sequences, and now this hemoglobin just can't function. We completely change its structure with one simple amino acid substitution. Lipids, nonpolar, with the exception of phospholipids. Phospholipids have 2 regions. They're polar and nonpolar. You might remember that term. It's called amphipathic. Here it is. Okay, and that refers to a compound that has both a polar and a nonpolar end. The majority of lipids are nonpolar. Okay, and they repel. Excuse me. They repel water. Okay, so fats, waxes, oils, most sterols, which are steroids, have single bonded carbons, which form unsaturated fats. And I'm sorry, saturated fats. If they have a double bond in there, it forms an unsaturated fat. So these are just some of the functions of lipids. Besides the plasma membrane, they can form other types of membranes. They can be involved in protection, insulation. They form the myelin sheath. Okay, and here's a typical phospholipid. Okay, it's got this polar phosphate head. Okay, there's the phosphate group with the charge on it. Okay, it's a negative charge here and a positive charge on this choline. And then we have the two fatty acid tails. Notice one is bent. That would be the unsaturated tail. Okay, unsaturated fats tend to form soft fats that are more flexible. Okay, like oils. And then we have this long saturated chain over here. Okay, which tends to form more rigid structures. The plasma membrane contains a saturated and an unsaturated fat, which allows it to be flexible and partially fluid, so that we can allow molecules that are embedded within the membrane to move around, and also it allows the membrane to move flexibly. Okay, this unsaturated and saturated fatty acid tail combo results in tiny openings in the membrane, which permits the diffusion of small non-charged molecules, things like oxygen, carbon dioxide, and nitrogen. Okay. The other thing about fatty acid tails is you notice they have lots of carbons and hydrogens. Okay. This is very stable. There's a lot of carbon, a lot of hydrogen. There are a lot of bonds in here. So there's a lot of stored energy. Okay. Lipids have the greatest amount of stored energy per mole compared to all the other organic compounds. And that's because of those very stable hydrocarbon tails that they have. Okay. Some of these tails can get quite long. They can be well over 20 carbons long. Okay, and again, the purpose of these is to provide stability and also to store energy. Okay, now again, for the phospholipid, here's our cell membrane, and we can see if we zoom in a little bit here, okay, that our cell membrane, the heads face outward towards the aqueous solution, the aqueous watery environment, the extracellular matrix in the cytoplasm, for example. But in the middle here, it's nonpolar. And that creates a barrier so that only small nonpolar substances can get through. Things like steroid hormones, estrogen, progesterone, testosterone, carbon dioxide, um, oxygen, nitrogen gas, things like that can get through. Larger molecules, things like glucose, have a much more difficult time getting through. Okay, they're too large, so they need to use carrier proteins or channel proteins in order to get them across the membrane. And then we have our carbohydrates. Okay, carbs, again, made up of monomers of glucose. Okay, glucose is immediately available for energy, to release energy to make ATP. Starch and glycogen are for stored energy in plants and animals, respectively. And cellulose is very rigid because it's made up of two different forms of glucose, the L form and the D form. You get this very rigid structure to it. It gets what's known as a beta linkage, and that makes for a very rigid structure, and that provides lots of strength and stability for plants. Okay, and the other key thing is the directionality. Okay, we kind of mentioned that before. Nucleic acids, they have defined ends with 3' and 5' ends. Okay, such as we see with DNA. Okay, and this is really important during DNA synthesis. Right, it's about the direction of transcription. Transcription always goes from 3' to 5'. And a new form of DNA is synthesized 5 to 3. Okay, we know proteins have an amino end and a carboxyl end, and that linear sequence of amino acids connected by those peptide bonds allows us to have that direction to it. And then, of course, we look at carbohydrates, and they have an orientation as well, especially when we look at L forms and D forms, okay, right-handed and left-handed forms. Just some other basic things. Make sure you guys are aware of the pH scale. Okay, you should know that it's logarithmic and that each time you change the pH, it changes by a factor of 10. So a pH of 3 is 10 times more acidic than a pH of 4. Okay. And then the other piece is bonding. Okay. Ionic and covalent are the strongest bonds. Ionic is the strongest, it's stronger than covalent. That involves electron sharing or electron transferring, as you can see, where hydrogen bonds are weaker. Right? Those are forces of attraction between a hydrogen and any negatively charged ion. Usually it's going to be between a hydrogen and an oxygen, or a hydrogen and a nitrogen. Okay, because oxygen and nitrogen have high electronegativities. Okay. That's as far as I want to go right now with this. We do have a little bit here. I'm just kind of scrolling down. Enzymes. We're actually going to address enzymes in another segment, but the key thing is that enzymes are catalysts that speed up the rate of chemical reactions, and they do that by lowering the activation energy, specifically finding an alternate pathway. Every enzyme has an active site, which consists of specific R groups where the reactions will occur. So these R groups can be hydrophobic, hydrophilic. The chemical nature and the chemical property of these R groups is going to determine how the enzyme reacts. We know enzymes can break down substances either through what's called catabolism, or they can build up substances by anabolism. So remember, anabolic is synthetic. That's to synthesize. So making DNA, making protein, making sugars in photosynthesis. Those are anabolic reactions. And catabolic reactions are things like cell respiration, okay, such as what occurs in the mitochondria, and digestion, hydrolysis. Okay. All right. So that's it for this first part. We don't want to jump on too many things, but that pretty much covers the basics of what you need to know for chemistry of life and the properties of water. And again, one of the things that they like to talk about, I think, are protein structure and cell membrane structure. And they like to have us come up with a way to explain how membranes form and what is it about membranes that's so important. So there's a tie-in between Unit 1 and Unit 2 regarding membrane structure and phospholipids. Okay, and again, it's these fatty acid tails and the phospholipids that dictate the permeability of the membrane, right? And then the other piece, of course, is with proteins. We're going to get into that when we talk about enzymes. That's in topic 2 that deals with photosynthesis, cell respiration, and cellular energetics. So I hope this helps you guys out a bit. Again, you have the answers here. You can always stop, rewind, and go back. So you can fill in the blanks on your own form on this. We do have a copy of it on Schoology as well. Okay, and that's it. So we'll see you again real soon. Thanks.